Hello everyone, in this video we are going to talk about effluent treatment plant, chemical biological treatment methods. So effluent treatment plant is designed to purify industrial wastewater for its reuse, and its aim is to release safe water to environment from the harmful effect caused by the effluent. Effluents from food and beverage factories contain degradable organic pollutants. Since industrial wastewater contains a diversity of impurities and therefore specific treatment technology called ETP. To put it simply, it is mandatory for all the food processing industries to set up ETP effective wastewater management. The ETP plant works at various levels and involves various physical, chemical, biological and membrane processes. Treat wastewater from different industrial sectors like chemicals, drugs, pharmaceutical, refineries, dairy, ready mix plants and textile, etc. Here in this flow chart, you can see different levels of stepwise processes of effluent treatment plant. The first is physical, or the preliminary treatment that includes screening, sedimentation, grip chamber and clarifiers. All of these processes are mechanical separation methods. The next is primary treatment that includes flotation, flocculation, coagulation and precipitation in the liquid waste. Then, you have secondary treatment that is also called biological treatment. In this process, water is treated with biological microorganisms such as bacteria, either in aerobic or anaerobic condition. The example are activated sludge process, aerobic process, trickling filter and anaerobic digestion. After that last is the tertiary treatment, which includes fluoridation, chemical coagulation and sedimentation, filtration, reverse osmosis and UV disinfectant. And finally you obtain the treated water. Now coming to the concept of different kind of solids present on wastewater. There are total solid, total suspended solid. Total solids include both the suspended solids and the dissolved solids, which are obtained by separating the solid and liquid phase by evaporation. Suspended solids are a combination of settlable solids and non-settlable solids, which are usually determined by filtering the wastewater sample. Next is volatile suspended solids, VSS. Biological solids that are volatile are generated during the aerobic/anaerobic treatment process and are termed volatile suspended solids. VSS solids remaining after evaporation or filtration are dried, weighed, and then ignited. The loss of weight by ignition at 600 degrees C is a measure of the volatile solids, which are classed as organic material. Next is fixed suspended solids, FSS. The remaining solids are the fixed solids, which are considered as inorganic mineral matter. The suspended solids associated with volatile fraction are termed volatile suspended solids, VSS, and the suspended solids associated with the mineral fraction are termed fixed suspended solids, FSS. The generation of biological solids, also referred to as sludge yield, is a result of the organic material BOB5 in the wastewater being converted to biomass, water and carbon dioxide. These excess biosolids generated during treatment can be thickened and ultimately hauled off-site, land applied or used as a compost amendment. In this diagram you can see how fixed suspended solids are filtered out from the wastewater in laboratory conditions. First, the water is dried to obtain dry suspended solid. Further, it is ignited at high temperature to give out termed fixed suspended solids, FSS. Now what are coagulants? So, coagulants are a substance which cause particles in a liquid to curdle and clot together. E.g. For reducing water turbidity, particles stay suspended in water rather than settling because they carry surface electrical charges that mutually repel each other. Coagulants carry the opposite charge to the particles and therefore cause the charge to destabilize when added to the water, resulting in the particles clinging together. There are two types of coagulants. Organic coagulants are generally used for solid and liquid separation and sludge generation, e.g. polyamines. Fat from dairy waste. Inorganic coagulants are particularly effective on raw water with low turbidity and will often treat this type of water when organic coagulants cannot, e.g. aluminum sulfate alum. Now look at this diagram to understand the process of coagulation. First, the coagulants are added to the water. The impurities in the water gets chemically bonded to the coagulants, forming the precipitate. These precipitate settles at the bottom after some time, leaving the clean water upside. After coagulants come the flocculants. Flocculants are substances that promote the agglomeration of fine particles present in a solution, creating a floc, which then floats to the surface, flotation, or settles to the bottom, sedimentation. This can then be more easily removed from the liquid. Flocculants can be organic or inorganic, and come in various charges, charge densities, molecular weights, and forms. Now look at this diagram to understand the process of flocculation. It shows the possible flocculation mechanism conducted by flocculant. Here you can see how neutralization of charge happens when a polymer is added to the colloids. At first, the small flocs are formed, followed by the larger flocs. Let's see different stages of flocculation. Stage 1. Suspended solid particles in wastewater are negatively charged. In the first stage of flocculation, a coagulant like aluminium sulfate is added to the wastewater. The positively charged coagulant molecules neutralize the negatively charged solid particles suspended in the water. Neutralizing these particles paves the way for them to flocculate together into a larger mass. Stage 2. The wastewater must be agitated with mixers. High-energy mixing is required initially to ensure that the coagulant spreads throughout the water. When flocculation is in progress, the mixing energy is reduced to prevent the mass of particles from separating again. Stage 3. Once floc is beginning to form, a polymer chemical is added to the wastewater. Polymers bridge the flocculant from micro to macro flocculant, meaning that the mass of particles collecting together gets bigger. This chemical also binds the collected mass together so that it does not easily disintegrate even when the water is slightly agitated. Stage 4. After flocculation is complete, the large solid masses can be removed from the wastewater stream. This is done either through settling where the floc drops to the bottom for removal or through the use of filters which capture the floc in the filtered material. Care must be taken when cleaning the filters to ensure that the phosphorus-rich floc is contained and treated. Next process is flotation. Process involved in removing the compound shows difference in wettability. Mostly used to remove hydrophobic compounds in wastewater, the flotation process operates by preparing a water suspension of a mixture of relatively fine-sized particles smaller than 150 micrometers and by contacting the suspension with a swarm of air bubbles of air in a suitably designed process vessel. Particles that are not wetted by water hydrophobic tend to be attached to air bubbles, levitate, float to the top of the process vessel and collect in the fourth layer. Difference in the surface chemical properties of the solids are the basis of separation by flotation. Surfaces that do not have strong surface chemical bonds that were broken tend to be non-polar and are not readily wetted, e.g. graphite and talc. These solids are naturally floatable. The wide application are separation of proteins from milk and clarification of fruit juices. The last process in the chemical treatment is precipitation. chemical process involves precipitate the chemically from dissolved substances in the wastewater by adding the reagent, which forms an insoluble compound with the to be separated matter. Positive ions such as heavy metals, but also negative ions like phosphates and sulfates, can be removed via precipitation. Precipitation occurs in the 1:1 mole ratio, in other words, one molecule of dissolved matter, for example SO42, present in the form of well-soluble natrium sulfate with one molecule of reagent, for example barium derived from soluble barium chloride, forms an insoluble precipitate barium sulfate in this case. However, a certain amount of overdosage is needed for complete removal. Coming to the biological treatment process. It is often called a secondary treatment process, which is used to remove any contaminants that left over after primary treatment. Chemical treatment of wastewater makes use of chemicals to react with pollutants present in the wastewater, and whereas biological treatment uses microorganisms to degrade wastewater contaminants. This treatment relies mostly on bacteria to break down unstable organic wastes using normal cellular processes to stable inorganic forms. Based on the process, biological treatment of wastewater methods are majorly classified into aerobic and anaerobic process. Aerobic process. Aerobic wastewater treatment is a biological process that takes place in the presence of oxygen. It is the rapid and the most efficient biological waste treatment, which remove up to 98% of organic contaminants. This process causes effective breakdown of organic pollutants and yields a cleaner water effluent than anaerobic treatment. Aerobic biological treatment processes include many processes such as activated sludge process, trickling filter, aerated lagoons and oxidation ponds, etc. Activated sludge process is the most widely used process for domestic and industrial wastewater. Aerobic biological treatment will remain efficient and stable in all conditions. Activated sludge process most widely used as biological waste treatment in secondary stage of wastewater treatment. An activated sludge process refers to a multi-chamber reactor unit that makes use of highly concentrated microorganisms to degrade organics and remove nutrients from wastewater to produce a high-quality effluent. In this method, the sewage containing organic matter with the microorganisms is aerated by a mechanical aerator in an aeration tank. This process speeds up waste decomposition. Aeration in an activated sludge process is based on pumping air into a tank, which promotes the microbial growth in the wastewater. Next in the category of aerobic treatment is trickling filters. Trickling filters (TFs) are used to remove organic matter from wastewater. The TF is an aerobic treatment system that utilizes microorganisms attached to a medium to remove organic matter from wastewater. This type of system is common to a number of technologies such as rotating biological contactors and packed bed reactors, biotowers. These systems are known as attached growth processes. In contrast, systems in which microorganisms are sustained in a liquid are known as suspended growth processes. This is a pictorial representation of trickling filter. Here you see the rotating effluent distributor that sprinkles the water through the bed made up of some media. The air is passed through the bottom of the chamber and effluent is collected after it passes through the aerobic bed. Now coming to anaerobic treatment process. This treatment process is effectively utilized to treat high-strength wastewater, and it employs organisms that function in the absence of oxygen, and it will typically treat high-strength wastewater to a level that will permit discharge to a municipal sewer system. Here, the amount of sludge produced is very small when we compared to aerobic treatment. Anaerobic treatment is a slow process and it occurs in many different stages. Anaerobic digestion is a biological process which is used in wastewater treatment plants for sludge degradation and stabilization. This process is accepted because it is able to stabilize the water with little biomass production. Biogas is produced as the bacteria feed off the biodegradable material in the anaerobic process. Overall, the process converts about 40% to 60% of the organic solids to methane CH4 and carbon dioxide CO2. An example of anaerobic reactor is UASBR, that stands for Upward Anaerobic Sludge Blanket Reactor. Here the sludge blanket is comprised of microbial granules 1 to 3 mm in diameter, i.e., small agglomerations of microorganisms that, because of their weight, resist being washed out in the upflow. The microorganisms in the sludge layer degrade organic compounds. As a result, gases methane and carbon dioxide are released. The rising bubbles mix the sludge without the assistance of any mechanical parts. Sloped walls deflect material that reaches the top of the tank downwards. The clarified effluent is extracted from the top of the tank in an area above the sloped walls. After several weeks of use, larger granules of sludge form which, in turn, act as filters for smaller particles as the effluent rises through the cushion of sludge. Because of the upflow regime, granule-forming organisms are preferentially accumulated as the others are washed out. Look at the diagram of USABR and you can observe that biogas moves upward. Another example of anaerobic treatment are anaerobic ponds. Anaerobic reactors UASB in tertiary treatment after the removal of suspended solids, oil and grease. Biological nitrogen removal is done by nitrification and denitrification, as well as biological phosphorus are removed. Let's understand the anaerobic degradation of organic matter now. For different phases can be distinguished in the overall conversion process of organic matter to biogas as hydrolysis, acidogenesis, acetogenesis, methanogenesis. So, this is the reaction sequences that happens in any anaerobic degradation. The suspended organic matter such as carbohydrates, proteins and lipids get hydrolyzed to break down to simpler compounds such as amino acids and fatty acids. From these simpler compounds corresponding acids are formed and the process is called as acidogenesis. After this, corresponding acetates are produced and the process is called as acetogenesis. Finally, in absence of oxygen, methane are formed and the process is called as methanogenesis. So just look at the overview of anaerobic degradation. The anaerobic biological conversion of organic waste to methane is a complex process involving a number of microbial populations linked by their individual substrate and product specificities. The product of one bacterium is often the substrate for others, and hence, a balance between the bacterial numbers and the substrate concentrations must be maintained. The first step in the process involves transformation of higher molecular mass compounds into compounds suitable for use as a source of energy and cell carbon hydrolysis. The second step, acidogenesis, involves the bacterial conversion of the compounds resulting from the first step into identifiable lower molecular mass intermediate compounds. In the third step, lower chain volatile fatty acids produced during acidogenesis are utilized by a group of bacterioacetogens to produce acetate. The fourth step, methanogenesis, involves the bacterial conversion of the intermediate compounds into simpler end products, such as methane and carbon dioxide. So, this was all about wastewater chemical and biological treatment. Hope you understood the concept.